can get some sense of if the protein was not there, not functioning. And you can see here, I guess, large data is in the eye of the beholder. So for modern day machine learning, this is like minuscule, like it's a little sneeze basically. And that makes machine learning really hard, actually. It makes it easy because things are fast and they're not unwieldy, and I don't need to worry about memory and compute time, but it's really bad for getting a good model. And that's actually one of the big challenges in this area, is there's very limited data at the moment. And I'll never forget one talk I saw years ago. And they actually, at one of the big machine learning conferences, they did this plot, and they basically showed the performance as you change through these different models. And you see this model is better, this model is better, this and this. And then they went to the next slide, and they said, okay, now we just made the data set 100 times bigger. And basically, that's so dominated the difference between the models that you're like, just get more data and don't worry about what what model. But when you're in this regime, you should care about the model. And to be fair, even in that regime, that was just one specific problem, but I think it kind of drives home a very nice point. And so, right, I mentioned that, different genes and different assays, like, kind of yield measurement in a different space. And I'm not going to, we have ongoing work as a sort of machine learning project on how to handle that in a rigorous, nice way. And in the meantime, we're doing some stuff like changing it to ranks and normalizing with the gene and stuff. If you want to know, we can go into this offline, but I'm not going to talk about that here. Right, and so now back to this sort of featureization, what we call featureization of a guide. So right, we have the 20-mer guide, and then a 3-mer after the PAM, and 4 before, and we need to convert that 30-mer nucleotides into something that's numeric, because any model I want to use in machine learning, fundamentally it assumes there are numbers there. They can be real value, they can be negative, they can be discrete, it doesn't matter, but they got to be numbers. And so, we're not the first people to have this problem. There's a rich history of computational biology and ways to do this. And so this, like we did, this is a pretty standard thing, but just to help you wrap your head around some of the ways in which we might want to do it. Imagine that, so a lot of the features we're going to use are based on the nucleotide content, and so what we're going to do is we're going to kind of make a dictionary where if there are 4 possibilities for a letter, then each of those basically gets, we call it one hot, because one of these bits is on and the rest are off, right? So this is the one that's hot, that's the one that's hot there, and it's called a one hot encoding, but I think you can see you just enumerate however many things you have, and then the code for it becomes however many there were. So here there's 4, which means there's 4 digits, and then you just kind of randomly give a 1 to A in that position, to T in this. And now what you do is when you want to convert this to a long numeric thing, first you look up the T. So you start with a T. So then you get 0010. Those are the first 4 numbers in the long numeric string. And then you look at the second letter, and it's a G. So then that's going to be followed by 1000. And you keep going like this. So that's going to give you some very long string, but we're not going to stop there. We're going to actually also do this for what we call order 2 nucleotides, and we get this sort of other kind of dictionary. You can do this up to order 3. You can do all kinds of other stuff that I'm not going to go into. I just want you to sort of wrap around in your head, how can you actually do this? And also the way I've just described this to you, like in this example, it's position specific in the sense that I start here on the left and I ask, was there a T? So this refers to the first position, and when I do this for every guide, it always refers to the first position, right? So when this is important in the model, it's because it's important in the first position. But I can also do things that are sort of position independent. So, and I'm going to actually do that not just for GC. I'm going to do it for every 2 pairs of letters, and I'm just going to say how many GCs were there, how many TGs were there, and things like this. And I'm just going to tack those on to this thing. This thing's just going to get longer and longer and longer, right? And this is where we need to be careful. Like, is it so long that I don't have enough data to actually deal with it properly? But in this case, like, even though they're really long, because so many things are zeros, it's actually not quite as long as we think it is in terms of the amount of data needed. And then something that comes out is important, and so we sometimes call John our Oracle featureizer, because John is, 1, he's smart, 2, he has very good intuition, and 3, he's up on a lot of literature. And so he said, there's crystallography experiments that show that, something happens at the 5-mer proximal to the PAM in sort of a cohesive way, followed by something over here and over here. So maybe when you're doing the thermodynamics, in addition to looking at the whole 30-mer, maybe also look at it just in these subportions and things like this. And so we interact with John, and we get features in this way as well. So we kind of use our rich history of just computational sort of an infinite amount of data, we could probably skip thinking through these things at all. Right, and so the last part, like I've now told you a little bit about how we do this, and you guys know more about this than I do, and so now the sort of last puzzle piece is what kind of model are we going to use? And so I've already told you a little bit about these regression trees, and so actually a lot of our CRISPR, these are just very, they're very rich models because you can get these interactions, right? If I split on this feature, let's say this is GC content, and then I go down to here and this is, let's say, A in position 1, then this is a very strong interaction between these two features because I have to go down this path before I get it. And so you can get these very complicated interactions, which is also why it's hard to interpret because you kind of need to like look at every interaction all at the same time to kind of understand it. So what we actually do is we actually use a bunch of those models together. There's a way to combine these models to do even better still, and this is called boosting. Does it have the name on here, boosting somewhere? Boosting. So boosting is a way where you take some sort of somewhat simple model. These aren't simple models, but we actually cripple them a bit. We keep them very, very simple, and then we apply this boosting technique. And so let me just explain to you intuitively what boosting is like. It's, I think, pretty easy to understand on an intuitive level. You take your data, and you fit the parameters on your training data, and then you go to your training data and you ask how well did this fitted model do on every one of my training examples. And then you weight each of your data points inversely to how well it did. In other words, if on that training point, after I fit the model, it did badly, then it's going to get up-weighted. And the stuff that it did, really well on is going to kind of fall out. It's going to get a very low weight. And then you reapply this learning algorithm, you refit another tree, and you add the results, then you start averaging the results from a sequence of these trees. And so there's a whole theory underpinning why you can do this, what that's actually doing, but on an intuitive level, that's all it's doing. And so it's, in some sense, it's refocusing its energy on getting those things that it didn't get right by subsequently adding in another tree. And these turns out to be very powerful models in a large number of domains. And so, in fact, that is what we use here.
You also do an iteration where you drop out a feature to see whether it made a difference.
Yeah, so I guess there's two things we do when we're doing things. One is we try to get the best model possible that we think will be the best, like for whoever wants to use it, we really think this is the best thing. And then another thing we do is we want to interpret it as best as we can. And so sometimes we drop out a feature because we think maybe it'll be better. But usually dropping one feature is not going to make a difference because if it's just one feature and we drop it out, it would have known it wasn't useful anyway, usually. Like, it could be that if you posited that there was just like millions of features and then you got rid of 500,000, that might help you if they were irrelevant. But if it's just one irrelevant one, it can figure it out, or else it's just doing a bad job anyway, basically. But then you might do it, like, we have some figures in our paper about here's the accuracy with all the features we finally used, and here's the accuracy if we pull this one out, to give people a sense of like, well, how important was it? But that doesn't necessarily tell you, actually. So it's actually more nuanced than that, because it could be that if I use this feature by itself, and we see this all the time. I think this happened with like microhomology, which we don't put in our model. If you put that in just by itself, it turns out to be quite or somewhat predictive. I don't remember how predictive this was. But if you take the model we've already settled on and you add it in there, it makes no difference, because things are, like, for example, nucleotide features cover GC content, and GC content is a proxy to thermodynamics, and you don't know what's a proxy for what and things like this. And so because everything's interacting, like, all you know is in the context of this model, if I pull it out, it doesn't change the performance. But that doesn't mean that it's actually not predictive, right? And then the flip side is, if I put it in by itself and it's not predictive, doesn't mean it's not predictive, because I could have put it in with a bunch of other stuff and together it might have been predictive. So this is why it's actually hard to do that, and it's also why it's hard to interpret. Yeah. But we do what we can in that vein to sort of poke and prod as best as we can.
To the previous slide, I want to make sure we got it right. Which is that sort of, we're having to go in because the data set is small and say, these are the features we hypothesize are important, and sort of give it a priori. And we might be leaving things out.
Absolutely. Yeah, completely.
What you're saying is that if we had a larger data set, we wouldn't have to be as got those features going into the model to start applying some of those more complicated things?
So yes and no. Yes, the larger the data, the more complicated